Hello friends, welcome back to Multi Question World. Today we are going to discuss some important and repeated questions of lab technician exam for various competitive exams. If you are a regular viewer of the channel, you can see that almost 99% of the questions we have discussed in the previous videos. You can watch the previous videos from playlist, and if you are watching without subscribing, please do subscribe the channel. Question number 1. The protein portion of the enzyme is option A apoenzyme, option B prosthetic group, option C cofactor, option D coenzyme. The correct answer is option A apoenzyme. The protein portion of the enzyme is called apoenzyme. Question number 2. Which of the following cells is not a WBC? Option A neutrophil, option B basophil, option C monocyte, option D thrombocyte. The correct answer is option D thrombocyte. Neutrophil, basophil and monocytes are white cells, but thrombocyte is not. So the correct answer is option D thrombocyte. Question number 3. The coloring agent used in anti-sera AB is option A methylene blue, option B aquaflavin, option C eosin, option D safranin. Which of the following coloring agent is used in anti-sera AB? The correct answer is option C eosin. The coloring agent used in anti-sera AB is eosin and the color is pink. Question number 4. Name the anticoagulant present in purple colored vacutainer tube. Option A heparin, option B EDTA, option C sodium citrate, option D none of these. Here heparin is present in green color vacutainer tube, sodium citrate in blue and EDTA is present in purple colored vacutainer tube. So the correct answer is option B EDTA. Anticoagulant present in purple colored vacutainer tube is EDTA. Question number 5. pH of buffered distilled water used in Leishman staining is option A 6.8, option B 7.2, option C 7.4, option D 8. The correct answer is option A 6.8. pH of buffered distilled water used in Leishman staining is 6.8. Question number 6. Blood collected in CPDA can be stored in blood bank refrigerator for dash days. Option A 35 days, option B 28 days, option C 21 days, option D 25 days. How many days we can store blood collected in CPDA in blood bank? The correct answer is option A 35 days. Blood collected in CPDA can be stored in blood bank refrigerator for 35 days. Question number 7. Hemolytic disease of newborn due to Rh incompatibility can be prevented by giving anti-D antibody to Rh negative mother within option A 48 hours, option B 72 hours, option C 24 hours, option D 36 hours. The correct answer is option B 72 hours. Hemolytic disease of newborn due to Rh incompatibility can be prevented by giving anti-D antibody to Rh negative mother within 72 hours. Question number 8. Which of the following is not a RBC diluting fluid? Option A normal saline, option B Daisy's fluid, option C Turk's fluid, option D Toyson's fluid. Here normal saline, Daisy's fluid and Toyson's fluid are the RBC diluting fluids and Turk's fluid is WBC diluting fluid. So the correct answer is option C Turk's fluid. Question number 9. During donor screening, persons having history of malaria should be avoided for a period of dash years. Option A 1 week, option B 2 years, option C 1 year, option D 3 years. During donor screening, how many periods should be avoided for a person having history of malaria? The correct answer is option D 3 years. A person having malaria should be avoided for a period of 3 years during donor screening. Question number 10. Bombay blood group persons have dash antigen in their blood. Option A antigen. Option B antigen. Option C recessive H antigen. Option D dominant H antigen. The correct answer is option C recessive H antigen. Small h antigen is the recessive H antigen and it is present only in Bombay blood group persons. Question number 11. Depth of improved Newbar counting chamber is option A 0.1 mm, option B 0.01 mm, option C 0.001 mm, option D 1 mm. The correct answer is option A 0.1 mm. Depth of improved Newbar counting chamber is 0.1 mm. Question number 12. Name the enzyme estimation done in semen sample to analyze the secretory function of prostate. Option A alanine transaminase, that is ALT. Option B aspartate transaminase or AST. Option C acid phosphatase or ACP. Option D alkylate phosphatase or ALP. The correct answer is option C, acid phosphatase. Acid phosphatase is the enzyme estimation done in semen sample to analyze the secretory function of prostate. Question number 13. The only cell in normal CSF is option A neutrophil, option B lymphocyte, option C monocyte, option D none of these. Which of the following cell is normally present in cerebrospinal fluid? The correct answer is option B, lymphocyte. The only cell in normal CSF is lymphocyte. These questions are familiar to you and you all knew the answer because we have discussed lot of times. Question number 14. Name the test used to detect bile salt in urine. Option A, Benzedine test. Option B, Faucher's test. Option C, Ehrlich test. Option D, Hayes test. The correct answer is option D, Hayes test. Test used to detect bile salt in urine is Hayes test. Question number 50. Ketone bodies in urine are option A acetone, option B beta hydroxybutyric acid, option C both 1 and 2, option D none of these. Ketone bodies are acetone, acetoacetic acid and beta hydroxybutyric acid. So, the correct answer is option C, both 1 and 2. That means both A and B. Acetone and beta hydroxybutyric acid. Question number 16. Normal sperm count is option A 80 to 160 millions per ml. Option B 40 to 80 millions per ml. Option C 100 to 120 millions per ml. Option D 140 to 200 millions per ml. The correct answer is option A 80 to 160 millions per ml. Normal sperm count is 80 to 160 millions per ml. Question number 17. Name the hormone detected in pregnancy. Option A ADH, antidiuretic hormone. Option B GH, growth hormone. Option C TSH, thyroid stimulating hormone. Option D HCG, human chorionic gonadotropin. The correct answer is option D, human chorionic gonadotropin, that is HCG. The hormone which is detected in pregnancy is HCG. Question number 18. Specific gravity of urine less than 1.010 indicates option A diabetes mellitus, option B diabetes insipidus, option C diabetic ketoacidosis, option D none of the above. The correct answer is option B, diabetes insipidus. Specific gravity of urine less than 1.010 indicates diabetes insipidus, that is heavy urination due to water and salt metabolism error. Question number 19. Glucose memory test is option A, glucose challenge test or GCT, option B, HbA1c, option C, GTT or glucose tolerance test, option D, PPBS or postprandial blood sugar. The correct answer is option B, HbA1c, that is glycated hemoglobin. Glucose memory test is HbA1c. Question number 20. Which of the following is not a renal function test? Option A, serum urea, option B, serum creatinine, option C, cystatin C, option D, total cholesterol. The correct answer is option D, total cholesterol. Total cholesterol is not a renal function test. All the others are renal function tests. Question number 21. Who is known as father of microbiology? Option A, Louis Pasteur, option B, Robert Koch, option C, Alexander Fleming, option D, Edward Jenner. The correct answer is option A, Louis Pasteur. Father of microbiology is Louis Pasteur. Question number 22. Serum electrolytes are analyzed using option A, colorimeter, option B, flame photometer, option C, spectrophotometer, option D, none of these. The correct answer is option B, flame photometer. Serum electrolytes are analyzed using flame photometer. Question number 23. Vandenberg reaction is used to detect option A, serum creatinine, option B, serum urea, option C, serum bilirubin, option D, blood glucose. Which of the following parameters we can detect by Vandenberg reaction? The correct answer is option C, serum bilirubin. Vandenberg reaction is used to detect serum bilirubin. Question number 24. Cobweb appearance of clot in CSF indicates option A, jaundice, option B, hemorrhage, option C, spinal constriction, option D, tuberculous meningitis. The correct answer is option D, tuberculous meningitis. Cobweb appearance of clot in CSF indicates tuberculous meningitis. Question number 25. Dash is otherwise known as bad cholesterol. Option A, LDL, option B, HDL, option C, VLDL, option D, chylomicrons. The correct answer is option A, LDL or low density lipoprotein is known as bad cholesterol. Question number 26. All of the following are motile bacteria except option A, Escherichia coli, option B, Klebsiella, option C, Vibrio, option D, Proteus. Which of the following is a non-motile bacteria? The correct answer is option B, Klebsiella. Klebsiella is a non-motile bacteria. All the others are motile. Question number 27. Oil and grease can be sterilized using option A, autoclave, option B, vaccine bath, option C, hot air oven, option D, none of these. Which of the following instrument can be used for sterilizing oil and grease? The correct answer is option C, hot air oven. Hot air oven is used for sterilization of oil and grease. Question number 28. Solidifying agent of a culture media is option A, peptone, option B, meat extract, option C, sodium chloride, option D, agar. The correct answer is option D, agar. Solidifying agent of a culture media is agar. 1 to 2% agar is used for a solid culture media. Question number 29. Dash is added to McConkie agar to inhibit the growth of gram-positive bacteria. Option A, bile salt. Option B, peptone. Option C, neutral red. Option D, agar. Which of the following inhibits the growth of gram-positive bacteria in a McConkie agar? The correct answer is option A, bile salt. Bile salt is added to McConkie agar to inhibit the growth of gram-positive bacteria. Question number 30. All the following are examples of transport media except option A, Venkataraman medium, that is VR medium. Option B, thioglycolate medium. Option C, Stuart's medium. Option D, Amis transport medium. The correct answer is option B, thioglycolate medium. Thioglycolate medium is not a transport media. All the others are transport medias. Question number 31. Which of the following tests come under IMVIC test? Option A, coagulase test. Option B, catalase test. Option C, citrate utilization test. Option D, none of the above. The correct answer is option C, citrate utilization test. IMVIC is indole, methyl red, Vogt-Bosker and citrate test. Question number 32. The type of immunoglobulin which provides local immunity is option A, IgM, option B, IgD, option C, IgG, option D, IgA. The correct answer is option D, IgA. IgA provides local immunity. Question number 33. Confirmatory test for AIDS is option A, Western blot, option B, Southern blot, option C, ELISA or enzyme linked immunosorbent assay, option D, CLIA or chemiluminescent immunoassay. The correct answer is option A, Western blot. Confirmatory test for AIDS is Western blot test. Question number 34. The bacteria which require factor 10 and factor 5 for its growth. Option A, Staphylococcus. Option B, Haemophilus. Option C, Brucella. Option D, Salmonella. Which of the following bacteria requires factor 10 and 5 for their growth? The correct answer is option B, Haemophilus. Haemophilus influenza requires factor 10 and 5 for their growth. Question number 35. RPR and VDRL tests are done for the diagnosis of option A, AIDS, option B, hepatitis, option C, syphilis, option D, malaria. Which of the following condition can be diagnosed by RPR and VDRL? The correct answer is option C, syphilis. Question number 36. All of the following are examples of blood parasites except option A, Plasmodium, option B, Brugia malayi, option C, Leishmania, option D, Giardia lamblia. Of the following, Plasmodium, Brugia malayi and Leishmania are blood parasites, but Giardia lamblia is not. So the correct answer is option D, Giardia lamblia. Giardia lamblia is not a blood parasite. It is an intestinal flagellate. Question number 37. The fixative used for blood containing specimen is option A, carnoise fluid, option B, 95% ethyl alcohol, option C, 10% formalin, option D, Zenger's fluid. Which of the following can be used for blood containing specimen? The correct answer is option A, carnoise fluid. Carnoise fluid is the fixative used for blood containing specimen. Question number 38. Staining technique for assessment of hormonal status is option A Pap stain, option B Shore stain, option C H&E stain, option D JSB stain. The correct answer is option B Shore stain. Staining technique for assessment of hormonal status is Shore stain. Question number 39. The word which refers to the closeness of a result on repeated analysis is option A accuracy, option B sensitivity, option C precision, option D specificity. In the previous videos, these terms are discussed in detail. The correct answer is option C precision. Closeness of a result on repeated analysis is precision. Question number 40. Mycology means the study of option A bacteria, option B immunity, option C parasites, option D fungus. For bacteria it is bacteriology, immunity, immunology, and parasites, parasitology. So the correct answer is fungus. Option D fungus is the correct answer. Mycology means the study of fungus. Question number 41. Infections induced by a physician is known as option A iatrogenic, option B nosocomial, option C congenital, option D subclinical. Which of the following term is used for physician induced infection? The correct answer is option A iatrogenic. Infections induced by physician is known as iatrogenic infection. Question number 42. The enzyme estimation done for the diagnosis of pancreatitis. Option A acid phosphatase, option B amylase, option C alkaline phosphatase, option D creatinine kinase. The correct answer is option B amylase. The enzyme estimation done for the diagnosis of pancreatitis